Hi, this is Alex from devstories.eu. AlphaFold Protein Structure Prediction, Episode 5 of 21. Real-world bioinformatics does not happen by manually clicking download buttons in a web browser. When you need to analyze hundreds of proteins, you build automated, scalable pipelines. That brings us to automating discovery, the AlphaFold Database API. You know the database holds millions of structural predictions, but viewing one protein at a time on a website is unworkable for large projects. If you're running a comparative analysis across an entire family of enzymes or screening targets for a drug discovery pipeline, you need those structures pulled directly into your computing environment. The AlphaFold database provides a REST API to solve this exact bottleneck. It allows your code to query the database and programmatically retrieve structural data without human intervention. The primary key for this automation is the UniProt ID. You can think of a UniProt ID as the universal barcode for any given protein. Your pipeline generally starts with a list of these IDs. Using a standard HTTP library in Python, you send a request to the API asking for the prediction data associated with a specific barcode. This is the part that matters. When you make that request, the API does not hand back a massive file full of three-dimensional atomic coordinates. Serving heavy files over a synchronous API call is slow and fragile. Instead, the API returns a manifest. This manifest is a lightweight response that describes exactly what resources are available for your requested protein. Conceptually, this manifest gives you the biological metadata and a directory of direct download links. It points to the actual 3D structure files in industry-standard formats. Crucially, it also provides links to the confidence metrics, such as the predicted aligned error, or PAE matrix. You need both. In an automated pipeline, downloading the structure is only half the job. You must also download the PAE data so your downstream algorithms know which parts of the protein are highly confident and which parts are flexible. Let us walk through how this logic flows in a typical Python script. You set up a loop to iterate over your list of UniProt IDs. Inside the loop, you make your network request for the first ID. Before doing anything else, you check the HTTP status code. Not every protein sequence in existence has a pre-calculated AlphaFold structure. If you request an unmapped protein, the database returns a standard 404 not found error. Your script must catch this error gracefully, log the missing ID, and continue to the next iteration. If you ignore status codes and try to parse a missing response, your entire batch job will crash on the first unsupported protein. When the request is successful, your script reads the returned manifest. You write logic to extract the specific download links you care about, which is usually the primary coordinate file and the corresponding error matrix file. Your script then makes secondary HTTP requests to those specific links, streaming the heavy data directly to your local storage or cloud bucket. By separating the query from the download, the API keeps the initial discovery phase incredibly fast. You can quickly check the availability of thousands of proteins, build a validated list of links, and then handle the heavy downloading in bulk. The real value of the AlphaFold API is that it removes the human bottleneck, turning a vast, static database into a fully programmable component of your research architecture. That is all for this one. Thanks for listening and keep building.